Welcome to Hard-Boiled Synthesis, Lecture 15. It's time to get our hands dirty. Today, I'm going to start computing effect sizes. I'm not going to do a lot of examples. I think I'm just going to process 3 studies just because I have 200 of these to do, and I don't think you guys want to watch me convert raw data into effect sizes 200 sometimes. But today should be an extension to the previous lecture about effect sizes. Now I'm just going to apply that discussion that we talked about of how do we quantify an outcome of a study. That's what we call an effect size. It's the data of meta-analysis, and really what it tries to do is provide a quantitative estimate of the magnitude and direction of an experimental outcome. Magnitude, so how strong was the effect? Was catnip that much more better than DEET? Right? Again, our goal is to test the claim of whether or not catnip is 10 times more effective than DEET. That's a magnitude, right? 10 times. And so by quantifying all the experimental outcomes, we could actually get to whether or not it's 10 times. The next part, which I didn't talk much about, is what other components does meta-analysis use to synthesize or aggregate study outcomes? And a key part of those stats is using the variance of effect sizes. And that's another thing that we need to compute, which will be used as the weights for meta-analysis. Meta-analysis is cool in that we don't assume each study is equal, just taking a regular average. What we do is we try to weight each study based on how we perceive that the results are sensitive to sampling error. And so studies with large sample sizes tend to get weighted more in the average than studies with fewer samples. That's a way to kind of adjust or account for the variability associated with sampling error. I'm running ahead of myself here. That's when I start talking about meta-analysis, I'll provide more insight on that. But right now, we need to start calculating the effects before we get to that final stage. I mean, holy smokes, it took 16 lectures before we get to the actual meta-analysis part. And so today, we're gonna have effect sizes calculated, and hopefully next lecture, we'll be able to actually test the claim. And so let's just get our hands dirty and start calculating effects. Now, I'm just going to give you guys a brief quick primer again on what the goal, what is the outcome we want to quantify from each study. And that is essentially this, a difference between repellency effects using catnip and a difference using repellency effects using DEET. Doesn't have to be DEET. Again, DEET is a positive control, a baseline for comparison. Many studies don't use DEET. They use nothing or a solvent. Now, DEET is kind of a solvent also. Don't need to get into those technicalities, but a non-repellent solvent. So like alcohol or acetone or something like that. Some sort of chemical that's used to dilute the repellent. Those are called negative controls, which are useful for what we want. But in the end, right, we're interested in testing the claim with DEET. And so everything that is the most useful for a meta-analysis involves a comparison with DEET. So this difference, right, for the majority of studies was essentially reported as a mean. So the mean repellency response of mosquitoes exposed to catnip and the mean repellency response of mosquitoes exposed to DEET, okay, the control. And we want the difference between the two. The difference is the magnitude. How different were they? and the direction. Was DEET, were mosquitoes more repelled by DEET than catnip? If you use this equation here, this difference, the value will be negative, right? So the opposite of the prediction that catnip is 10 times more effective than DEET. If catnip is more effective than DEET, then the difference will be positive, which will be in favor of the claim that catnip is better than DEET. Now, there's more to this than just the difference, right? All the studies measure and report things differently. They may be reporting landing times. They may be reporting concentration of optimal repellent use to scare off mosquitoes. They may measure contact responses versus spatial responses. There's a whole many different ways in which you can measure repellency effects. And so we need a common currency. We need to standardize the difference. That's what this is. That's the homogenized variance that we use to standardize all the effect sizes. And this is what we're going to call delta. This is our effect size. This is what we're going to compare across all studies. That standardized difference. And so this is what we're going to calculate. And I'm not going to use anything fancy to calculate these things. I'm just going to go into Excel and write the equations out so you could see exactly what's being done. And then we'll compute some effects. Now there's more to it than this, because the homogenized variance, this orange symbol here, has its own equation. And it's not a pretty equation. Just between you and me, it's not really doing anything elaborate. What it's doing, it's taking the standard deviations from the control and a treatment mean, converting them into variances, adjusting those variances using Bessel's correction. Variances tend to have a slight bias in estimating the population variability. Averages those two variances and then converts it back into a standard deviation. Okay, maybe it is kind of like a lot of acrobatics going on here, but the goal is to like homogenize the two variances. Key assumption here is you assume that the variance of the control mean and the variance of the treatment mean have similar variability, right? That's why you're pooling them. You're trying to improve the way you standardize the difference by averaging those two variances. Basically, you have two replications of that variance estimation. And so why not make use of it to improve how you standardize the effect? That's an assumption. Most people don't test whether or not there's a difference between the variants. If the variances differ between the treatment and control group, then technically you should not be homogenizing. They're heterogeneous. They're not homogeneous. And so you can't homogenize those variance components. We're not going to look at that. We're going to skip that. That becomes part of the, not necessarily deviations from best practice, but more like overlooking assumptions of effect sizes. And then finally, we're going to calculate the variance of the delta, the variance of the effect size, which also looks like an awful equation. And this equation is assumes many things, which are not important to you. I would love to get into the details of this. I spent a lot of time trying to figure out how this variance equation is derived like 10 years ago. I figured it out. It was cool. It was like certainly a feather in my hat to figure out all these derivations of all these equations. But it's nothing that you need to know other than it's a mishmash of things that aims to estimate the variance of the effect size. We need this for meta-analysis because these become the weights. These become the weights in which we measure individual effects when we come to average all the studies. On top of that, we're going to correct both the effect size and the variance with this thing called a small sample adjustment, J, as it's known in the literature. This corrects for some of the issues associated with this difference type, standardized difference type effect size when it tries to estimate an effect. When the sample sizes are small, when a study has a small sample size, the effect may be an overestimate of the true underlying effect. And J here, this small sample bias correction, shrinks that estimation. You know, you may get a huge effect estimate based on a study with small sample sizes. This aims to kind of like shrink that overall effect, so it's not overestimating. This is a type of bias that occurs. I don't know what else to say to this other than this is a standard practice to adjust the effects. These are things that are well studied, well known. And so, yes, there are many layers of corrections, but these are justified because we understand under certain conditions, this is not these equations don't quite work the way they should. And so you need these corrections baked in to help improve inferences at the meta-analysis level. If all your effects have an overestimated bias, that means your overall pooled aggregated effect will also have this overestimated bias. This may make it more difficult to properly assess true and null effects because there's a bias that occurs in your estimation, and you don't want none of that stuff. And so this is these things are a necessary component, even though algebraically they're confusing and ugly. We need to do before we even do meta-analyses. All right, so let's just do it. Now my goal now is to like transcribe these equations in Excel by hand, right? I'm just going to type out the equations just so you see what gets done. Let me bring up Excel here. All right, so basically we're going to do 3 studies, right? So I got the study 2, 24, and 29, all of which were studies with some variation of catnip. There are many effects per row. Each row is an individual effect, right? That's something I designed from the start to make it easy for me to calculate effects. But it means there's repetitions, right? Because what's happening is studies report multiple types of controls, right? They may report a positive control and report a negative control, but you just have the one catnip estimate. This is called a common control problem. I'm not going to account for that, right? If you were serious about meta-analysis, you would try to account for that shared information between effect sizes. That shared information, what it does is it creates the illusion that you have full replication across effects. But in reality, you need to adjust your model to account for that shared information. That's beyond the scope of this class, this course. If you're interested more in that kind of stuff, contact me. I could talk for days about these kinds of things. But the goal now essentially is to calculate delta, the effect size, calculate its variance, and then correct those things to create Hedge's D and the variance of Hedge's D. Again, in the previous lecture on effect sizes, there's some confusion in how these effect sizes are described in the literature. In some disciplines, this would be known as Hedge's G, but I like to keep things close to the bedrock, to the source. Larry Hedges called it D, and that's what it's described as in his original publication in 1981. To me, D, you could backtrack. If you use the word D to represent the effect size, you could backtrack to the original source and see how it was derived. If you use G, it's hard to kind of validate or identify the source because Larry does not describe it as D in the manuscript, in the publication. Okay, I'm talking way more than I need to. I just need to transcribe these equations. And because everything's by row, things should be straightforward. Now, there's already some issues with the studies in that some of the controls used did not report any variability. But I'm still going to transcribe the equations, and we're going to see what happens when you throw in a bunch of zeros into those equations. Usually that's not a good thing. Okay, so hopefully you can see this. Let me double check. You can see it. Excellent. Let me make this slightly bigger. Okay, so the goal now is to start computing these effects. I'm just going to do it little chunk by little chunk so that you see what gets calculated with each level in the effect size estimation. So first thing we want is the delta, which is just the difference between the means reported in the study, right? So we want response mean minus control mean. So that is the delta. And so we already see negative values means DEET is performing better, or the control is performing better than catnip. And just eyeballing all these effects, you can see that a bunch of them are indicating that the control is performing better than catnip. Now, I didn't tease apart whether or not these are positive or negative controls, but by the most part right now, the effect sizes are negative, indicating that catnip is not performing as good as the control. Okay, so the next part is the standardization of delta, which is the homogenized variance of the treatment and control mean. And so that's what we need to plug in here. It's a huge equation. I think I have it out for you guys. There it is. Okay, so this is the ugly part. I may do some transcription errors here. We start off with the square root. And then there is a division between the two. I don't know why I'm talking through this. It's divided by the degrees of freedom. Minus 2. I got my notes here on the side because I don't really know these equations by heart. You'd think after all these years I do, but I don't. Right, now throw in the sample size of the treatment. This is basically the Bessel correction. Minus 1, and then multiply these by the variance, which is the square root of the standard deviation. Does that work? And we'll call that sigma. And see if these are calculated. See, all right. So hopefully you could see what happened here. We got a bunch of null. Yeah, you can see. So already there were homogenized variances that we can't estimate because of all the zeros. And what's happening here? Okay, no, not because of the zeros, because I have question marks. Now, in study 24, I remember this. Study 24, it wasn't described whether or not the variance, the bars around the mean reported in the figure, was a standard error or standard deviation. Nowhere in the manuscript does it ever state what it is. And so I kind of have a rule for these situations. Pick the more conservative of the two. The more conservative, if it's a standard deviation or a standard error, pick the one that provides the broadest thing. That way you kind of baked in some of the error associated with the effects size because of the lack of reporting, is you're assuming the more conservative effect from that. And so I'm not going to assume that the standard deviation, these are standard deviations. I'm going to assume that they're standard errors, and that they need to be, I'm going to assume that they're standard errors and they need to be converted to standard deviations. What happens with that is the numerical value extracted becomes bigger, and therefore there's more variability associated with the effect. And so the goal now is to convert these into standard deviations. I'm assuming that they're standard errors, convert them into standard deviations. And the equation for that is fairly straightforward. The standard error is the standard deviation divided by the square root of the sample size. And so you throw the square root of the sample size towards a standard error. And so the standard deviation is the standard error times the square root of the sample size. See, super easy. Actually, I have a book chapter which I describe all these equations, and I get that equation wrong. I have that equation wrong reported. The most simplest of conversions in the entire chapter, and I got that equation wrong. Okay. So we want to convert 5 here. I'm going to get rid of my shorthands here, because it was unclear to me what the identity of these variances were. And then I'm going to estimate, convert these into standard deviation. So we got a square root of the sample size. Right? So these values are going to become much bigger now. These are going to become 9s instead of 3s. Again, I'm baking in this challenge that I have in the effect by assuming that there's more variability. Assuming of the two to pick, I'm picking the one that introduces the most variability. And then finally, here we go. So there, now these are corrected. And we were able to estimate a sigma from these things. Okay, so now we want to standardize the delta. Delta stand for delta standardize, and it's basically just delta divided by sigma. Right, so we weren't able to standardize a bunch of them because we have 0 variances in what was reported in the studies. So a bunch of these effects are can't estimate an effect size. And let me just quickly look to see what effects are the weird ones, and I can see right now they are the ones. Okay, so it's not so bad. They're the negative control effects. So here they use copper tone and some other lotion. instead of DEET. The DEET one, we were able to compute an effect, but the copper tone and the lotion didn't work. And the reason for that is there was no repellency effects with the positive control, with the negative control. This is the challenge with negative controls, is that you're going to get little to no variability in outcomes, which makes it difficult to do a comparison. It's great if you're like trying to compare whether or not a repellent happens. It will almost always repel something compared to nothing, right? That's that's kind of like that's a BS test in my books. Is like, you got nothing versus a repellent. Does it repel? Well, yeah, it's always going to repel compared to zero effects. But nonetheless, in terms of like our meta-analysis, this isn't a big deal, because those were negative controls, which are not necessarily what we're interested in. We're interested in comparisons with DEET. But I'm going to keep them as uncalculable, because I like to conserve all these details in my analyses, and later I could describe in my Prisma plot that 75 or so outcomes could not have an effect size estimated. All right, more to do. We did the delta, we did the sigma, we did the standardized the delta. Next is to estimate the variance of delta, the variance of the effect size. This too has a cute equation. And the way I have this equation is slightly different from what is typically reported in textbooks. I don't use the abbreviated conversions of the sample sizes. That's just some rearrangement of algebra that happened at some point when Larry wanted to describe the effect sizes. It's totally unnecessary. And then we want to take the square of the delta. Now, common criticisms with the variance of delta is that it includes delta in the estimation. To me, that's not a big deal, because delta is a non-central t distribution. That's its underlying distribution. To estimate a mean and the variance of a t distribution, you have to include the mean into it. That's a property of the distribution itself. So to me, this equation, even though it seems like, oh, you're recycling information, and sure, it's not an ideal plugin using the estimated thing back into the equation, it's formulated in a way that makes sense statistically, because it's a reminder that, yes, it has a t distribution. And then there we go. We got the variance. Now let's quickly zip through the variance of each thing and have an eyeball of which studies are going to get weighted more heavily than others. Now, the real bummer about repellency studies is they're heavily under-replicated. I mean, sample sizes could be from 3 to 10. I don't think I got any study that uses a sample greater than 10 to estimate a mean. This is not ideal at all. But what can you do? I mean, we're not the ones who did the experiments here. We have to make use of what's available. Luckily, we have this weighting scheme that at least tries to weigh studies, but if everything has low sample sizes, the weighting really doesn't mean much. But we still do it. Okay, so we got the variance. Now, finally, we're going to correct the delta, correct the variance, and then we'll finally have our effect sizes. So this will be called D, and then finally var D. And these two columns here is what we're going to plug into the stats for meta-analysis. Oh, but I kind of jumped ahead. We need to estimate J, which is the bias correction. And this too has kind of like a weird equation to it. It's a abbreviated form of a non-central bias associated with a non-central T distribution. You know, back in the day, everyone was calculating the stuff by hand, right? This was presented in 1981. Not everyone had personal computers back then. Oh, maybe if you were an academic, you probably had a personal computer at this point. And so you could calculate this stuff. But a lot of these things needed to be derived in a way where you could use a pen and paper to figure out. And the best equation to estimate J uses estimates from a probability distribution function. That's not something you could do in a hand. I don't know if you're old enough, like I am. When I was learning stats, we just look up these giant tables in the back of stats books to find values associated with degrees of freedom and probability distribution stuff. And so it becomes tedious. And this equation here is a shorthand version of approximation of that nice equation. So now when you use software to do this, chances are they're probably using the beautiful version of the bias correction rather than this abbreviated version. All right, almost done. I messed up. Again. All right, let me see what I did here. Not 48. 4 times this. There we go. How? Why is this not calculating? Okay. Again, still, what is happening here? There we go. Okay, so here are the biased adjustments. Let me check to see if you can see that. I'm always worried that you can't see it. You can see it. Right, forgot to put up the equation. So that's what I just totally transcribed in Excel. Right, so because all these studies are have such small sample sizes, the bias correction is fairly big, from 5% to 7% downward adjustment of the effect, right? So for example, the effect for this first one, which has a negative value, DEET is performing better than catnip. That magnitude of effect is going to, we're only going to take 95% of that value, right? We're removing 5% of that effect. Let's just do that math right now. Right, so instead of 3.5, we have 3.33. We reduced the bias. And so all of these effects are going to experience significant reductions because they're based on really crappy sample sizes. And then finally, let's end it with the D, which has a slightly different equation. which is J squared. This 2 gets adjusted downward. And there we have it. We have our effect sizes for 3 studies. These were straightforward because they were all reported in means. Other studies I'm going to have to convert from odds ratios because they only report counts. But you saw me write all the equations. I don't know how long it took me, but it's doable by hand. I often get emails from people saying, hey, do you have tools to estimate effects? And I'm always a little confused by that because the equations are readily available everywhere. But I could see that you could be concerned because there's a lot of goofy acrobatics. If you're ever stuck, like this case here, calculating by hand, I suggest you verify your equations using canned software. There's a ton of canned software that estimates the effect. I think there's a bunch of online stuff that calculates the effect for you if you want. And so just verify. See if you've written the equations properly before you go on to your meta-analysis. So I'm not fully confident that I properly coded the equations here. And so I'll double check to see if I got these done. Again, a reminder, a bunch of these effects I could not estimate because there was no variability in the control. That's the nature of these negative controls, is you may get 0 means from the outcome. That's not useful at all. I mean, it's not even useful to do reporting regular stats on that stuff. You couldn't do a proper t-test comparing the means if you have just a flat out 0 included in that. All right, well, there you go. I feel like I'm going to do this now, behind the scenes, calculate all the effects. Next lecture, I think we're finally going to be able to answer this question. Next lecture. Huh? We're no more... no more of this. Right, we're going to be able to ask the question whether or not catnip is 10 times more effective than... I just need to... dirty and actually start estimating the pooled effect, the average effect across all studies. That's the sweet stuff right there. All right, I totally talked longer than I should have. Happy New Year's. Have a good break. You know, take it easy. All right, see you later.